just make sure that, yeah, okay. So I know how to work with you. Okay, can you all hear me? I'll just move this up. Is that better? I think that's better. Okay, so it's my pleasure to present an update on the development of Takeda's recombinant tetravalent dengue vaccine candidate. And in my presentation today, I'm going to cover the vaccine construct, preclinical safety, the preclinical immunology and efficacy, cellular immune responses in non-human primates, cellular immune responses in humans, and then I've got a few slides to describe our clinical program and some of the results that we've seen so far, and then I'll finish with a summary. So, Takeda's live attenuated tetravalent dengue vaccine candidate contains the pre-membrane and envelope genes of each of the 4 dengue virus serotypes, shown here, expressed by the attenuated dengue virus 2 genome. And I'm sorry, I don't see the red dot very well, but it's the upper line thing. Throughout my talk, you'll see the abbreviation TDV, and this is the nomenclature that we are going to use for the vaccine from now on. In the past, we did talk about the vaccine as Denvax. We're no longer using that name, so just to be clear about that. And where you see TDV2, well, of course, that's the monovalent dengue vaccine 2 candidate. So the vaccine induces neutralizing antibodies to all 4 of the dengue virus serotypes. The vaccine also induces a multifunctional and cross-reactive cellular CD8 T cell responses to the proteins of the encoded by the dengue virus non-structural genes. Now, the vaccine is stably and sufficiently attenuated. The virus genome has been fully sequenced, and the mutations that are necessary and sufficient for the attenuation of the vaccine have been identified. And these mutations are shown, the position of these mutations is shown by the red triangles in the first slide. There are 3 mutations that are necessary and sufficient for attenuation of the vaccine. These occur in the 5' non-coding region, and in the coding regions for the non-structural proteins 1 and 3. These mutations work individually and synergistically to produce a stably attenuated vaccine, and this has been demonstrated both in vitro and in vivo. The vaccine is safe and well tolerated in mice and non-human primates, including when administered by different routes of administration and at higher doses than will be used for human vaccination. Studies so far show no evidence of neurotoxicity, nor are there any significant effects on body weight, temperature, or hematology in non-human primates. The vaccine induces neutralizing antibodies to all 4 dengue serotypes in mice and non-human primates, and protects these animals against challenge with wild-type dengue viruses of all 4 serotypes. In addition, AG129 mice immunized either with monovalent dengue virus serotype 2 vaccine or tetravalent vaccine are protected against challenge with a lethal dengue serotype 4 virus. So showing the cross-protective effect of the monovalent dengue 2 virus vaccine candidate. Now, we've completed many challenge studies in non-human primates using different formulation ratios, different routes of administration, and different vaccine schedules. these have demonstrated the protective effect of the vaccine against viremia in non-human primates. In addition, these studies have provided evidence of sterilising immunity against dengue virus 2 and against viremia following challenge with dengue virus 4, even in the presence of low antibody titers. And for those of you who've seen some of our clinical, some of the immunology from our clinical studies before, you'll know that the lowest antibody responses are against dengue virus 4 with our the current formulations of our candidate vaccine. So looking at cellular immune responses in non-human primates, a recently published study that investigated the immunogenicity and protection of a rapid vaccination schedule using different routes of administration in non-human primates provides evidence that the vaccine induces a multifunctional and cross-reactive CD4 and CD8 T-cell response with the production of interferon gamma, interleukin-2 and tumor necrosis factor alpha, targeted at the dengue non-structural proteins. And this study demonstrated that the vaccine protected animals against viremia following wild-type dengue virus 2 challenge with no increase of neutralizing antibody titers post-challenge. This study then provides evidence that the vaccine elicits innate immune responses thought to be important for the development of an adaptive immune response. Now, the importance of innate immunity, and in particular the protective role of CD8 positive T cells during viral infection is probably well established. Protective mechanisms, of course, include direct cytotoxicity and the production of pro-inflammatory cytokines such as interferon gamma and tumor necrosis factor alpha. And it is clear that the innate immune response is largely responsible for the clearance of primary dengue virus infection. There's also evidence to suggest that both CD4 and CD8 T cells may contribute to protection against homologous reinfection or heterologous dengue infection. And for more discussion on this, I refer you to the papers by Daniela Weisskopf in the last year. So we analysed the kinetics of the CD8 T cell response to vaccine in flavivirus naive human volunteers in terms of cytokine profile produced and the non-structural proteins recognised. We found that the vaccine elicits CD8 T cell responses targeting dengue virus 2. These cells produce both interferon gamma and tumor necrosis factor alpha predominantly. The cytokine responses were highest 90 days following primary immunisation and still detectable after secondary immunisation. And the vaccine-reactive CD8 T cells were both multifunctional, producing 2 or more cytokines simultaneously, and they cross-reacted to the other non-structural proteins of the other 3 dengue virus serotypes. Clearly, these are results from a phase 1 study. There were only a few subjects in this study, and we will be exploring further the cellular immune responses throughout our clinical trial program moving forwards. This slide shows an outline of our completed clinical studies. So far, we've completed 4 phase 1 studies in adults from non-endemic regions using different formulations, different routes of administration, and different schedules for the vaccine. In terms of phase 2, we've completed 1 phase 2 study, and this was conducted in adults and children. in dengue endemic regions, so including both subjects who were either dengue seropositive or seronegative at baseline. This slide summarises the safety from our phase 1 studies. These clinical trials provide evidence that the vaccine is well tolerated in both high and low dose formulations and with different routes of administration, either subcutaneous or intradermal administration. There were no serious related adverse events. There were few mild or moderate systemic events. These were mostly headache, nasopharyngitis, nausea and myalgia most commonly. Importantly, there was no constellation of symptoms suggestive of dengue fever, and all grade 3 and 4 adverse events were not related to the vaccine. In terms of local injection site reactions, these were mostly of short duration and more common with the intradermal route of administration, and we will not be continuing with our investigations of the intradermal route for our vaccine. There were no meaningful changes in blood chemistry or hematology, so no significant neutropenia or thrombocytopenia, and no discontinuations due to adverse events and no deaths. So this slide shows the primary objectives for our phase 2 study, the one phase 2 study that we have completed. These were to investigate the safety and tolerability of the vaccine administered subcutaneously to healthy adults and children, and to investigate the immunogenicity of the vaccine against all 4 dengue virus serotypes. This study was conducted in dengue endemic countries, so in Puerto Rico, in Colombia, Singapore and Thailand, and this was a randomized double-blind placebo-controlled study. So this slide shows the study design. There were 2 parts to this study. The first part was an age de-escalation, starting with adults 21 to 45 years of age and finishing with young children. The second part of the study was an expansion phase where we vaccinated 200 subjects, 200 children, 18 months to 11 years of age. 2 doses of vaccine were given 90 days apart, and the formulation that we used in this study was our high-dose formulation. In terms of safety, what we saw was the same as what we'd seen in the phase 1 studies. There were no discontinuations due to adverse events, no related serious adverse events. In terms of systemic events, these were mild to moderate in severity and self-limited. Again, headache, nasopharyngitis, nausea and myalgia were most common. All grade 3 and 4 adverse events were not related to the vaccine, and there was no constellation of symptoms suggestive of dengue fever. Injection site reactions again were mild to moderate and of short duration, and there were no deaths in the study. So this slide shows the GMTs in vaccinated subjects. You can see quite clearly that the vaccine induces an antibody response to all 4 serotypes, and this response with this particular formulation of vaccine is highest against dengue virus 2, and the antibody levels are lowest with dengue virus 4. And this slide shows the antibody response for vaccinated subjects who were either seronegative at baseline on the left-hand side or seropositive at baseline on the right-hand side. And I think you can see that quite clearly that the antibody response is strongest to all 4 serotypes in subjects who are seropositive at baseline. We also believe that these results suggest that a second dose may add very little to the overall effect of the vaccine. This slide shows the proportion of subjects who are seropositive to multiple dengue viruses at the different time points. So the blue bar is the proportion who are seropositive at baseline. The red bar is the proportion of subjects seropositive after a single dose of vaccine, and the green bar is the proportion of subjects who are seropositive after 2 doses of the vaccine. And I think you can quite clearly see that more than 80% of subjects were seropositive to all 4 serotypes after 2 doses of vaccine. And greater than 90% of subjects were seropositive to at least 3 serotypes after just 1 dose of vaccine. And this slide shows the proportion of subjects seropositive to dengue, to multiple dengue viruses, stratified by age. The trend, the association between baseline seropositivity and age is quite obvious in this slide. I think it's also interesting to see that in young children, 90% was seropositive to at least 3 serotypes after a single dose of vaccine. And now I've come to the last 2 slides, the summary slides. And I think I'm probably well within time. So in summary, we've completed 4 phase 1 studies, and we have one phase 2. We've got 1 phase 1 study ongoing. I believe next month we start another phase 2 study. In terms of safety, we've vaccinated more than 600 subjects with our vaccine now. All of these dose formulations were well tolerated. Injection site reactions were of short duration and mild to moderate in severity. Systemic adverse events were mostly upper and lower respiratory tract infections. There was no constellation of symptoms suggestive of dengue fever, and there were no related serious adverse events. The vaccine induces neutralizing antibodies and levels of seroconversion greater than 80% to all 4 dengue serotypes after 2 doses. We see also evidence that the vaccine seems to prove to induce levels of seroconversion greater than 90% to at least 3 serotypes after a single dose. The vaccine elicits multifunctional, cross-reactive cellular immune responses in humans, and these are thought to be associated with protection against dengue. Data I didn't show suggests that the response to dengue viruses 1, 3 and 4 are homotypic responses and not just a heterotypic part of a heterotypic response to dengue virus 2. And finally, our pivotal efficacy study is in preparation at the moment, and we plan to start this next year. Thank you. I'll take any questions. Oh, sorry. I should thank Dr. Macareo, please.
Thank you very much. Swadika. Welcome, everyone. Thank you for this opportunity to speak this morning. We always present a little disclaimer that I speak for myself and not for the U.S. Army as a whole. But I will also disclose that our organization and our parent organization work very closely with Sanofi Pasteur, as well as the NIH and Takeda and GSK. So our interest is in producing a viable vaccine for the world, and we're happy to collaborate with any organization that has a viable product. So today I will be speaking about the NIH vaccine, but we do work with others. So one question that might arise in your mind is, why in general develop a live attenuated vaccine? Both the Takeda product, the Sanofi product, and the NIH product are all live attenuated vaccines. We've seen success with other flaviviruses, yellow fever, and Japanese encephalitis with live attenuated vaccines. And I won't read my slide to you, but essentially, with other vaccines that are on the market that are live attenuated vaccines, you expect to induce lifelong immunity, and they're very highly immunogenic, even after 1 dose. And you can see the other live attenuated vaccines that exist. And except for those that are administered by mouth, generally speaking, they're a 1 or at most 2 dose vaccine. And that's important, and I think in a situation like dengue, where there would have to be broad vaccination and expense associated with it in many countries that don't have a lot of resources. This is the overview of where we are. These are the 3 of the 4 candidates that we will be speaking about this morning. You can see the Sanofi Pasteur vaccine is based on a yellow fever background, chimeric. And then what we just heard about, the Takeda vaccine based on a dengue 2 background with a chimeric presentation as well. The NIH vaccine is a little bit different. What has been done, and I'll go into a little bit more detail, is that full-length viruses have been used with a deletion that has resulted in a attenuation, except that you can see that for dengue 2, that is a chimeric. I'll explain more briefly. Just to give you an idea, as we just heard, there's a lot of data behind the data, and I won't present everything, but suffice it to say that a lot of candidates were looked at before they were distilled down to the components that we see in the current NIH 2 vaccine products that are available to be evaluated. So you can see on the right-hand side what the components that we've ended up with. This is a closer look at the vaccine itself. So for dengue 1, 3, and 4, or let me say for dengue 1 and 4 serotype that's in the vaccine, a deletion of a 30 nucleotide deletion in the untranslated region of the 3' end was done. And the studies that were done early demonstrated that this resulted in sufficient but not over attenuation of those virus strains. For the dengue 3, it wasn't quite sufficiently attenuated, and there was an additional deletion done. And for dengue 2, the simple 30 nucleotide deletion did not result in sufficient attenuation. This may not actually be because the virus is too strong. Actually, the strain that was used was taken from an outbreak that presented mildly to begin with. So it may be just that this deletion did not further attenuate it, and then seeing the difference between deletion without the deletion was just not possible. But in any case, what was done was to essentially create a chimeric from the dengue 4. What we've ended up with selecting to move forward are 2 vaccines that have been both show significant promise. We call them TV003 and TV005. The only difference between these 2 vaccines is that in the dengue, the TV005, there is a 1 log higher amount of the dengue 2 component. But the dengue 2 component is the same as in the dengue, the 003 vaccine that I just showed the components of. It's just that there's a little bit more dengue 2 in the 005. I won't go through all the studies that have been done. Many of you are probably familiar with this work, but just to give you an introduction, initially naive subjects were studied, and PRNTs, plaque reduction neutralization titers, were done early and then in an extended period. Most important thing, safety. Bottom line of this slide is that there really isn't much to say. The only common and relatively significant finding is that a rash occurs in a large percentage of the vaccine recipients. And as it turns out that this rash tends to correlate with a tetravalent or response to all 4 serotypes to the vaccine. If you don't have a rash, you may still have a tetravalent response. But if you do have a rash, it's highly predictive of a tetravalent response. So we've almost come to think of this as a good thing. And I'll show you why I say that, because not only is it predictive of the tetravalent response, but it's a very, very mild rash. In fact, subjects that have it often do not think that they have a rash on their diary cards, but the rash is only seen by investigators who are actively looking for it. So what you can see here is the rash is not the little red papules. The rash is just this mild discoloration that you see all throughout the field. You can see here, like this, very mild. By comparison, this is a regular wild-type dengue rash. So possibly related, but significantly different in the amount. Looking at both of the vaccines in this kind of naive study, we see that there's a fair percentage of people that respond with viremia, indicating some replication of the vaccine, but it's relatively low in both vaccine candidates, and it does not last very long. That's the take home from this slide. When you look at the plaque reduction neutralization titers from the initial views of 28 and 42 days, you see that the dengue 2 component is kind of bringing up the rear, much like the Sanofi vaccine is the one that we saw the least respondents in. But the 005 a little bit better. If you extend out something also similar to the Takeda data a little bit longer, you see a significant response across the board. The percent that seroconvert on the extended 003 goes from 50% to 76% for the dengue 2 component, and remains high with across the board. And for the dengue 5, even better. So really, I would direct your attention to the bottom left line. We're looking at significant responses to all 4 serotypes, greater than 90%, with the 005 vaccine after 1 dose in naive subjects. This is the 003, at least a 74% trivalent or greater response after 1 dose. And you see the 005 product, greater than 90% trivalent or greater response with 1 dose, and high 90s for tetravalent response after a wait. So from that data, again, much like Produce, and I appreciate that you gave this presentation earlier because it makes things easier. The question is 1 dose or 2 doses. The current most highly advanced in development product in terms of phase of development is the Sanofi product, and that's a 3-dose vaccine. We've seen from participating in those trials, and that's not a minor undertaking. So for us, we look at this data. You can see this is the 003, the yellow writing in the top left corner. And we go 6 months, if we give a booster at 6 months, you see that the percentage that respond, it does increase. But what you see is a very, very small increase in mean peak titers. You would expect to see, if there was a true boosting effect, something like a 4-fold increase, and we see very little. And again, much like the data previously presented, in the 2nd dose, we don't see viremia or rash. And with the dengue 5 component, it's even more pronounced in terms of those that seroconvert. So you see that after the 1st dose, 92%, 97%, 97%, 97%, there's really not much additional gain with that 2nd dose. You see the full change after the 2nd dose is very minimal. So I think as Dr. Plunkett asked the question before, there's some suggestion that, well, maybe you give that 2nd dose a little too early, that's why you're not seeing a significant boosting effect. So what if we wait 1 year? So the study was done, waited 12 months before giving the 2nd dose, and you can see really it's the same story. Very little boost with the 2nd dose, no viremia, very little rash. And additionally, you can see that the titers remain relatively unchanged over the course of that year. So what we can say, thus far, is that it looks like the vaccine produces nearly sterilizing immunity at 12 months with a minimal boost in antibodies or titers. That's not good. Okay, so just to summarize the data between the 2 vaccines, Both of the admixtures are safe and well tolerated in studies that we've seen thus far in naive subjects. There's been additional work in the, flavivirus experienced subjects. But we can see across the board that 005 appears to be a little bit better in terms of the immunogenic response. And it appears that the effect of this vaccine goes out for at least 1 year in terms of the immunogenicity. So currently, the NIH's position is that whether it's 003 or 005, it would likely go forward with a one-dose strategy. This is now just looking at all the vaccines, except for the last one that we presented, but the 3 from Sanofi, Takeda, and NIH. I previously listed Takeda as a 2-dose vaccine, so maybe that's not accurate. Yeah. Okay, we'll leave it there for now. I don't want to misspeak on behalf of you. But anyway, you see fairly good responses across all the vaccines, truly. But the question that plagues all of us is that there is no good correlate marker for protection. We can't say that you reach a certain antibody titer or a certain PRNT response, and that this means that you will be protected against wild-type virus, and that's really the major problem. So no one really knows until you do the phase 3 trial and you really put it out there. And we know that the Sanofi Pasteur vaccine has been put out there, and has been moderately effective. Not as much as hoped, but it is the one that has gone the furthest. So what NIH wanted to do was to get a little bit better idea of how the vaccine may actually work in the real world. And they created a challenge study. And if you may recall that the strain the attenuated strain that's used in the trial is a chimeric from the dengue 4. What subjects were challenged with was this dengue 2 deletion 30, which was not quite as attenuated as the vaccine virus strains, and not from the same original source. So it's a different dengue 2 that subjects were going to be exposed to. And you can see that with, give you more of the, yeah. So you can see that actually, following delivery of this product, that all subjects get viremia and a very high frequency of rash. So what normally happens it being exposed to this vaccine. And this is what happens when we give it versus 003. So what was done was 003 vaccine was given, and then 6 months later, they were challenged with this dengue 2 strain. No viremia at all. So what we can say, from this limited study, is that the 003 vaccine provided 100% efficacy against this specific monovalent dengue 2 challenge against viremia and rash. That strain didn't really produce disease, vascular leakage, fever, etc. So it's hard to comment any further than that, but we can see that it protects against viremia and rash. So here we see a little bit more of the details. If you go out to day 180 and look at the titers, you can see that in the ratios, that response to the placebo is huge. But in the challenged subjects that received 003, very little change in titers. Also supportive of its efficacy. Currently, let me just go back to this. So that's where things stand right now with the vaccine. NIH has pretty good confidence going forward based on these results and this success of the monovalent challenge that they've undertaken. And there's been a lot of interest from others. These are some of the people that they're working with to license the product, exclusive and non-exclusive. What's interesting to us here and what we're involved with is that we have already screened, and we will begin enrollment this coming weekend on a phase 2 trial that will be done at Pamukuk Lao Hospital. And we will be using essentially both of the vaccine products, 003 and 005, in adults, and then giving them vaccinations, and then a month later evaluating where they stand. If both vaccine products look safe, but one appears to be a little bit more immunogenic, then that is the one that we will go forward with. And then when we go forward, we will enter an age de-escalation trial. And there's a similar trial that's planned also in Brazil, not exactly the same format, but essentially another phase 2 trial to evaluate the vaccine in the Americas. That's all I have to present. Dr. Weerachai, sitting right here, Dr. Silak, they will be the heavy lifters in doing the trial. Dr. Weerachai is the principal investigator on the trial that's started now at PMK, and we look forward to great success. Thank you.
Thank you, Ajahn Chukian, for the opportunity to present our efforts in Thailand on the development of recombinant dengue vaccines. And basically what I've tried to present to you is mainly preclinical studies or basically immunogenicities in mice and monkeys. And I've tried to tell you that we have tried to develop a number of dengue vaccine candidates. That the two major approaches that we took are the chimeric-like attenuate vaccine candidates and chimeric virus-like particle, and lastly our efforts to combine these two candidates into the LAV prime and VLP2 strategy. Now, reason why we take too much pain to try to do this arises some years ago when I went to a vaccine meeting in Europe. I sat next to Steve Whitehead, and I asked him, why should we try to develop other dengue vaccines when Sanofi and Pasteur's vaccine has been so well advanced? And he answers that, we don't know whether Sanofi would succeed. And maybe now our efforts basically arise from collaborative network funded by the National Science and Technology Development agencies in Thailand. And these comprise several collaborating centers. I just mentioned Medical Biotechnology Unit of National Center for Genetic Engineering and Biotechnology, our group in Chiang Mai University, and we have collaborated with Center for Vaccine Development in Mahidol University on laboratory testing and small animal studies. And also in our monkey works, we collaborated with Institute of Molecular Bioscience, Mahidol University, who work in collaboration with AFRIMS in working in rhesus macaques, and with Bogor Agricultural University in working with Cynomolgus macaques. And I'd like to start with our live attenuate vaccine candidates. And basically this is based on a strain called 166.13 This is a laboratory strain we generated in our laboratory from a full-length sine clone of the prototypic dengue 2 virus, 16681. And this is a virus from DHM grade 4 patients in Bangkok back in 1964. And it's been propagated in the laboratory and constitute one of the most lab-adapted strains. Now, the strain that we are using carries 3 attenuating mutations from strain 16681 PTK53. That's a vaccine candidate generated in Center for Vaccine Development of Nopporn University. And based on the work in US CDC, it has been well characterized that PTK53 actually carries these three attenuating point mutation in the 5' UTR, NS1 and NS3. And to generate this, we just substitute these three nucleotides in the wild type 16681 full-length DNA clone with the attenuated mutations from PTK53, and that makes 16681 3PM. Okay, so basically very similar to the Takeda Dengue parent strains, but that Takeda strains is actually these full-length derived from full-length DNA clone of PTK53, so quite similar. Now, this is just to tell you that we used the biotechnology techniques in generating the virus from full-length DNA clone that we prepared based on a plasmid, P blue script, and that can be grown in E. coli, and manipulation of this full-length DNA clone can generate chimeric or mutant strains in the laboratory. And once we have 16681 3PM, we generate chimeric virus employing PRM and E sequences of dengue 1, 2, 3, and 4. So basically, all of these chimeric vaccine candidates will be chimeric viruses. Now, we try to generate chimeric LAV virus by using recent clinical isolates. Okay, so our PRME sequencer we used for making chimeric sets of tetravalent vaccine candidates are based on recent clinical isolates from Konkan or Songkhla in Thailand around 2003 and 2005. And when we introduced a chimeric PRME sequence into 16681 3PM, and we tested on the immunogenicity in mice and in cynomolgus monkeys, we found that not all of the introduced PRME sequence would be immunogenic. So we have to improve on immunogenicity studies by several methods, and that includes using PRM and E sequences from wild type donor that we know based on infection in cynomolgus macaques to be immunogenic, or we use consensus sequence, and in a number of vaccine candidates, we intentionally adjust the PRME sequence that we introduced, or we also try to adapt the virus in vivo cell by limiting passage in vitro. Now, this summarizes for you that after several years of working in the laboratory, this constitutes the tetravalent chimeric LAV that we generated, and this is the serotypes of the chimeric PRME sequence, and the donor strains, the source patients that we derived the strains. So basically, viruses donors were from 2003 or 2005 epidemic period in Thailand, and they are either from DF, DFJ, or dengue. And we don't really have a specific reason for selecting these donors. Basically, we try to find what we would call regular isolates, okay, from the patients. And then we find that certain PRME sequence is not so good in stimulating neutralizing antibody response. So we have to change the PRM donor, and for dengue 2, we need to, at the end, use consensus PRME sequence, and also we need to adjust PRME sequence in the laboratory also. Dengue 4, we need to do adjustment by limited passage in vivo cells in vitro. Now, for those that we do not do sequence adjustment, that means that it is particularly dengue 1. It is immunogenic in monkeys right away, so we don't have to do further adjustment. Now, this table also shows you that we have also modified PRM junction by introducing mutations into this junction. The reason why we did this is because of the recent findings that we learned by characterizing particles of dengue, particularly dengue 2, and from recent immunology studies of infected persons, particularly in Thailand and also in the other parts of the world. Now, what I try to tell you is that if we grow dengue, particularly dengue 2, in vitro, and we look at the particles of this virus, we found that they are not homogeneous. Actually, particles of dengue or flavivirus need to undergo maturation events during the export. And we found a number of years ago that dengue 2 particles are not mature in total. So we found that if we grow dengue 2 in mosquito cells, particles represents a mixture of immature, partially mature, and mature particles that are different in the surface texture, and that result from difference in conformation and oligomerization PRM proteins. And maybe because of this heterogeneity in particle structure, infected person generate a lot of cross-reactive anti-PRM antibodies, and these anti-PRM antibodies do not neutralize dengue virus in vitro. In contrast, they enhance dengue virus infection of leukocytes that express Fc gamma receptors. And so infected person generate antibodies that can both neutralize and enhance virus infection, at least in vitro. And what is quite surprising is that if you look at antibody against dengue virus and look at anti-dengue antibody that's recognized ED3 domains, and that particular domain is of interest because it is receptor binding domain, and it is found that only minority of antibodies in infected person recognize these important domains. So infection by dengue virus may not give purely neutralizing antibodies, but they can also elicit infection-enhancing antibodies, and all the neutralizing antibodies, only minority recognize receptor binding domains. So this summarized for you that dengue virus particle populations are a mixture, and we can see mixtures of mature, partially mature particles and mature particles, in addition to virus-like particles generated from infected dengue-infected mosquito cells. And this slide shows you that the arrangement of PRM and PRM here and E in immature particles is quite different from that of mature particles. Right? And in particular, what I would like to direct your attention is that your attention is that the ED3 domains, that is binding to receptor, is kind of obscure in immature particle, but they are exposed in mature particle. So particles are not the same, and maybe they can elicit different types of antibodies in infection. And the work from our lab published 4 years ago, we categorized these dengue-2 virus particles in collaboration with Richard Kuhn in Purdue University, and we show that it is possible to modify the ratios of different particles by simply change sequence just N-terminus to PRM cleavage junction. And showing this table, if we modify the sequence at this particular level, we can reduce the proportion of mature particle and increase, at the same time, increase the proportion of immature particle. We modify slightly different sequence at this particular junction. We can enhance the proportion of mature particle and at the same time reduce the proportion of immature particle. So we can basically alter the proportion of different particles of a virus population. So the idea is that can we improve the vaccine efficacy at the end by immunizing persons with LAV virus with high proportion of mature particle, or similarly immunize with VLP with high proportion of mature particle, and also do the AFRIMS vaccination. So what we try to do to show the proof of concept is to what we try to do to show the proof of concept is to compare two different LAV. CD1 3PM is regular chimeric dengue1 PRME sequence under dengue2 166 stage 1 3PM backbone. Okay. And to compare with this one, we generate CD1 4PM with an enhanced PRM cleavage. Okay. We also generate virus-like particles based on dengue2 with no cleavage, okay, and with enhanced cleavage. And we study the properties of these two approaches. This is shows you the results of the LAV. CD1 3PM has something like 76% PRM cleavage in viral cells, 42% in C636 cells, and we introduced one point mutation in PRM junction that enhanced cleavage to almost 90% in viral cells, 80% in C636 cells. So we make LAV that are different in the level of maturation, and that enhances the specific infectivity of the virus about 5-fold. But even with this enhancement in virus infectivity, this 4PM and also 3PM does not cause virulence in newborn mice. Okay. They are immunogenic in adult mice and rhesus macaques. They have reduced replications in mosquitoes. Okay. These are criteria that are used to assess live attenuated dengue vaccines. Okay. So basically they perform quite similarly. And then we do a monkey trial, rhesus macaques. The 3PM and 4PM do not protect against infection. And that's not surprising. Now, what we found is that if we look at the viremia of the challenge virus, during immunization and challenge. Okay. And one thing I would like to mention is that in the challenge, we try to use recent dengue isolates as a challenge strain instead of the prototypic strains. And you can see that these dengue 1 or dengue 2 challenge virus, these are recent clinical isolates, and they generate viremia in 100% of this small number of rhesus macaques. We immunize 6 with 3 p.m. and the other 6 with 4 p.m. Okay. There don't seem to be much difference in terms of viremia during the immunization. But in the challenge with either dengue 1 or dengue 2 challenge strains 60 days after the immunization, we see that we still see number of monkeys with viremia with 3 p.m. group, but we don't see any viremia in the 4 p.m. group. So that seems to suggest, based on this limited number of rhesus macaques, that immunization with chimeric dengue virus with enhanced PM cleavage results in the reduction or absence of viremia of recent dengue isolate during a challenge in rhesus macaques. So it seems that by introducing PM cleavage mutation seems to confer some benefits in the challenge model in rhesus macaques. Now, when we do modifications in LAV, we cannot make LAV without maturation because it won't grow. Okay. We also, when we do modifications in LAV, we cannot make LAV without maturation because it won't grow. Okay. We also cannot make LAV with 100% PM cleavage because it's dependent on the purine cleavage activity of the cells. So we cannot promote the cells to do 100% cleavage. So we try to do VLP because we generate this by introducing only 2 viral protein PRME into cells, and that number of cells can be used to express PME, but mammalian cells will die because of overexpression of PME, so we switch to insect cells, and in this particular experiment, C636 cells. And if we select cells with high expression, we can try to purify virus-like particle by autocertification and demonstrate by electron microscopy that in fact the particles formed based on the expression of PRME alone in selected cells. And by introducing mutations into PRME cleavage junction, we can make a number of preparation. I show you this preparation with native viral sequence, and this gives us 24% PM cleavage. This with cleavage inhibitory mutation that gives us 4% cleavage. This one with cleavage enhancing mutation that gives us something like 83%. And in other experiments, we can do something like 100% cleavage VLP. And they look quite different depending on the maturation level. This is Y-type VLP, VLP without cleavage. And these are all small VLP with the size of about 26 to 28 nanometer in diameter. The one with enhanced cleavage has also elongated forms in addition to spherical forms. Okay. So this one has 80% pure cleavage. Now, if we immunize mice with with VLP, and this is the one with enhanced cleavage. We see that they are not very immunogenic in mice with 1 or 10 microgram or even 20 microgram purified proteins. Okay. We need to use adjuvants, and it seems that quite a number of adjuvants work well in mice, boosting the neutralizing antibody response and to this VLP in mice. And if we compare the two forms of VLP in mice, we found that in the absence of adjuvants, VLPs with enhanced cleavage appears to be more immunogenic. In VLPs with enhanced cleavage appears to be more immunogenic in eliciting PRNT antibodies, okay, in mice. Okay. But when we use strong adjuvants like Abisco, the differences between the two groups seem to be not as high as in the absence of adjuvants. Okay. But there's some tendencies of higher neutralizing antibody response in mice with VLP with Abisco. And at the end of the two doses that we use to immunize mice, okay, we see in the presence of Abisco, when we see quite similar neutralizing antibody levels in mice, we also see that there are cross-reactive antibody responses to dengue1, dengue3, and dengue4, and quite similar to dengue2. There seems to be no major differences in neutralizing antibody titers. Now, if we look at ED3 binding antibody activity in mice, two groups of mice at week 20, okay, quite surprisingly, we see that if we normalize dengue2 binding activity and look at cross-reactive binding activity against dengue1, dengue4, and dengue3, we see that immunization with VRP with high cleavage of PRN resulted in comparatively higher level of cross-reactive anti-dengue1, dengue4, and dengue3 EDT recognizing antibodies in the mice. So it seems that by introducing cleavage modulating mutations into the VLP to generate VLP with different levels of maturation, the differences in virus population can be recognized by mice, and there appears to be fine differences in the antibody generated in mice against these VLP preparations. So the question is whether this VLP would be immunogenic in monkeys, and we did these experiments in cynomolgus cakes, which are not as efficient a model in producing neutralizing antibody as rhesus macaques. And I show you these summary results with PBS control followed by VLP, okay, to show you that VLP with enhanced cleavage at an arbitrary dose of 10 microgram dengue2 VLP doesn't do anything to the monkeys, even with strong adjuvants. Okay. So it seems that we probably need higher dose of VLP, which may not be economical. So we try to do experiments using VLP as a boosting immunogens, following LAB that is not so immunogenic. Okay. So these green lines here show the GMT genic. Okay. So these green lines here show the GMT following infection with a LAV, okay, one-time symptom PFU of CD chimeric dengue 2-3PM, and this is derived from NS1-123. That is not so immunogenic, and you see it's very low anti-response observed after one injection during the 3 months period. If we boost these sets of monkeys with 10 microgram VLP with Apisco, we see that there's a lot of increase in anti-response in 2 and 4 weeks after the boosting effect of dose of VLP. And this level is quite similar to what we observed after 2 doses of recent isolates infection in Cynomolgus macaques. Okay. And if we do the experiments boosting anti-response following heterologous LAV, the chimeric dengue 1-3PM, we don't see much increase, okay, in these orange lines of the GMT using the same dose at the same time of dengue 2 VLP. So it seems the VLP can boost antibody response following homologous LAV immunization. We tried to develop chimeric ED3 VLP, okay, by substituting domain 3 of VLP with dengue 3 sequence, okay, and the rest of the backbone is dengue 2. And testing this also in the prime boost model, again following very weak dengue 3 LAV, and we see that if we inject dengue 3 LAV, chimeric LAV, this particular strain is not immunogenic, okay, very low anti-response. After we boost it with VLP, this chimeric ED3 VLP, we see quite dramatic increase in anti-response, okay, in both monkeys that has some NT with very low undetectable anti-response. Okay. So again, we see that the GMT are now reaching the level that we see with wild-type recent isolates infection of monkeys. So the ideas that we are pursuing can be summarized in our pilot studies in monkeys that CDN1 LAV with enhanced peer cleavage provides good protection against anemia during the same wild-type virus challenge in monkeys. And in the presence of adjuvants, dengue 2 VLP, and also in limited studies, chimeric dengue 3 VLP will enhance peer cleavage, boost anti-antibody response in homologous LAV prime monkeys to the levels that are comparable to those seen in wild-type virus infection. And that gives us a cause to pursue further. I'd like to thank our collaborators in Department of Microbiology, in Biotech, in Concord University, in Research Institute of Health Science in Chiang Mai University, our collaborators in CBD, particularly Dr. Suti Yoksan, who performed all the NT works for the monkey studies, and also to Dr. Nopporn of Institute of Molecular Bioscience of Nopporn University, who organized all of the monkey studies. That's quite difficult work for her to do. And I thank you for your attention, and I'm happy to take questions.
Someone, asking what's the difference between CYD24 and CYD14 or 15? That's the 15 will be given by Dr. Nalaya. I just want to mention here that the sample size of the 2B CYD23 based on the assumption the vaccine efficacy more than 0. But for the CYD14, assumption based on the vaccine efficacy more than 25%. So the sample size is larger. So these are the countries that involved in this trial, including Thailand, Malaysia, Indonesia, Vietnam, and the Philippines. Philippines account for the majority of the participants. So altogether, 10,600 participants were screened, and 10,275 were randomized. But however, with various dropped out in between, we had altogether 6,800 received vaccines and 3,400 received placebo, that's the control. And per protocol analysis, we have 6,710 in the vaccinees and 3,350 in the placebo group. The demographic data, the mean age is around 8.8 years. And take a note here that the majority, about 67.6%, has the baseline anti-dengue infection. And the anti-JE seroprevalence was 52%. And the seroprevalence for the flavivirus, 78% for the total. So when we look at the primary endpoint, the efficacy as shown here, that when we look at the intent to treat, that is in the orange or yellow, is 54.8%. And in the per protocol analysis was slightly higher, 56.5%. And when we look at the prevalence, the incidence of the total of the dengue cases reported in this study, the total cases detected is 595, okay, and divided into various serotypes. When we look across various countries, Thailand, so in Thailand, during the 1 year of the active surveillance, the majority was serotype 2, similar to Indonesia. But however, in Malaysia, is nearly equal of the 4 serotypes. And the Philippines, I forgot to point out that the majority was serotype 4. So when we look at the subgroup analysis for each serotype, for the dengue 1 is the efficacy around 50%, for the dengue 2 around 35%, and dengue 3 78.4%, and for the dengue 4 around 75.3%. What I read here is all the per protocol analysis. So when we look at the critically important reduction in severity of the disease and hospitalization, I think it's very important. It's important to look at the vaccine efficacy in terms of lessened severity. So these vaccines show in the subgroup analysis show that the vaccine efficacy against dengue hemorrhagic fever cases of 88.5%, with the confidence interval of 58 to 97.9. And for the hospitalized cases was 67.2, and with the confidence interval of 50 to 78, so it's quite high. And when we look at closely with the age group and serology at baseline. We found that this slide shows the proportion of naive versus immune anti-dengue subjects by age group. So noted here that the higher the baseline of the against the dengue infection, the higher the efficacy. The same thing that the vaccine appear to be higher in terms of efficacy in a higher age group. Okay, so in summary, this slide shows trends for variable efficacy by age, and age is a biologically plausible surrogate for baseline dengue immune status. In terms of safety, side pain was the most common solicited reaction reported, around 30% in both groups. The majority were mild, appeared within 3 days, and disappeared also within 3 days. The solicited systemic reaction, malaise was the most common reported symptom, around 30%, 29 in the vaccinees and 23 in the control. And altogether, 647 reported serious adverse events. There were no difference between two groups, and no immediate hypersensitivity reported, no case of visceral trophic or neurotrophic disease. So in summary, the CYD tetravalent manufactured by Sanofi Pasteur appeared to be safe, moderately efficacious, with 81% reduction in risk of severe dengue infection. However, many questions remain to be answered. It is necessary to do long-term follow-up to assess duration of protection, to assess long-term safety, and also to assess the impact on the dengue transmission. The second point of concern is what is the epidemiology threshold of the dengue activity upon which the national dengue vaccination program are justified, and the cost-effectiveness of the program, as the vaccine is probably not going to be inexpensive and should only introduced in a high-risk group or in a higher age group with a highest vaccine efficacy. So these are the questions, and maybe some other more questions remains to be answered. Finally, I would like to acknowledge and thanks especially for the participants and community of the Kampang Phet and Ban Pong and Potharam, the AFRIMS Biological Unit, Sanofi Pasteur, Faculty of Tropical Medicine, Mahidol University, Ministry of Public Health, and Kampang Phet, Ban Pong, and Potharam hospitals, and especially for all the due diligence staff of Kampang Phet, Ban Pong, and Potharam, and including Ajahn Siraks and all senior staffs that sitting around here. Thank you for your attention.
Thank you, Professor. Please.
Thank you. First of all, I'd like to say thank you to Professor Rani for inviting me to be the speaker today and share interesting data with all of you today, and for the nice introduction from the chair. So today I will be talking over this topic as mentioned by the chair. So this will be next to follow up from the Professor Pani on CYD 14, this will be 15 in Latin America results. So scope of the presentation today will be on Sanofi Pasteur dengue vaccine candidate. This data as mentioned will be on CYD 15, so that's Latin America and the Caribbean. The main result will be reviewed here. Phase 3 efficacy study, then I'll compare the two studies, CYD 14 in Asia, PAX, and the CYD 15 in Latin America and the Caribbean. Result the first 25 months, that's the active phase of these two studies, and the overall conclusion. So this is actually the same as Professor Pani. So I just go through very quickly the key points I would like to stress out is Sanofi Pasteur live attenuated dengue vaccine used recombinant technology with the backbones of yellow fever 17D vaccine strain. It is the most clinically advanced dengue vaccine in development. And these slides just want to stress out that Sanofi Pasteur has established a robust clinical trial program which includes more than 40,000 individuals, nearly 30,000 of whom have received the vaccine. So this slide will just show that countries that included in the phase 3 efficacy study. You can see that these are all among the 10 highest dengue endemic countries as reported to the WHO. And the country included in the 2 phase 3 efficacy studies. So from Asia Pac in CYD14 will be Thailand, Malaysia, Indonesia, Vietnam, Philippines, as mentioned earlier by Professor Pani. For the CYD15 efficacy study in Latin America and Caribbean, will be Mexico, Honduras, Colombia, Puerto Rico, and Brazil, in which I'll go through the result with you. So these 2 studies have been published. For CYD14, it's been since July of this year in Lancet, and for CYD15 in the New England Journal in just early last month, November this year. So we're going through, I just walk through the result of the CYD15, which has just been published in the New England Journal of Medicine last month. This slide is trying to show you the CYD14 was conducted in 5 countries in Latin America and the Caribbean. These countries have some of the highest incidence of dengue in the world, as mentioned earlier. So this is just want to tell out it's the same study, the same kind of study designs and sample size as CYD15. So it's an observer blind, placebo-controlled, multi-center phase 3 trials into which over 20,000 sub-children and adolescents aged 9 to 16 years were randomized into the study. The eligible children were randomized assigned 2-to-1 ratio, received 3 doses of dengue vaccines and or placebo at 0, 6, and 12 months. And follow-up in the study was conducted in 2 phases. The active phase, which is from which the current data set to be shared with you today, and the ongoing hospital phase, which is ongoing right now and for about 4 years. So it'll be up to 6 years of this total study. So this is study designs and the sample size of the study. So participants in CYD15 were divided into several subsets of different analyses. The 2 efficacy populations were defined to compare the numbers of dengue cases between the vaccines and the control group. For the per protocol, outcome will span from 28 days after the 3rd vaccination, that is month 13, was through the month of 25. This will be the per protocol where we find the vaccine efficacy. And for the intention to treat group will be all the participants who receive at least 1 dose. So we start from day 0 when they enter the study into month 25 during the active phase. And we have a subset of the around 2,000 subjects, participants, which have been in the immunogenicity and reactogenicity subset. Roughly about 20% of the total participants. So go to the key results of the study. So this is so there are the per protocol and the intention to treat. For the per protocol population, vaccine efficacy was 60.8%. And for the primary endpoint, which met the primary endpoints, which you can see from the 95 freedom confidence interval is 52 to 68. And the key for the primary endpoint analysis was confirmed as in the intention to treat analysis as well. We show among all participants who received at least 1 dose of the dengue vaccine with at least one injection was 64.7, with the confidence interval of 58 to 69%. So both confirm the same results. This is the secondary objectives of the study. We show the serotypes specific vaccine efficacy, which can be observed during the 25 months active surveillance period, including the 12-month post-dose free per protocol assessment. Serotype-specific vaccine efficacy was observed, ranging from 50.2 in serotype 2 to 80.9 for serotype 4 in intention to treat analysis. So you can see that efficacy was higher in participants with pre-existing dengue neutralizing antibodies than in those who were seronegative. As the same study, which is consistent with CYD14, the same results. So clinically important reductions in severe dengue and hospitalization were also observed in the study. You can see from this slide that the vaccine efficacy against dengue hemorrhagic fever, which is a severe form of dengue infection, due to any serotype is 95% from the intention to treat analysis. And the vaccine efficacy against hospitalized dengue cases due to any serotype is 80.3% in subjects received after at least one dose of the CYD vaccine. The difference in efficacy by countries are likely due to differences in the baseline antibody levels and serotype circulations. So in this case of Mexico, the local dengue baseline compared to the other 4 countries show that the baseline seropositivity rate combined with circulation are probably the main result. Mainly, so you can see from the study in Mexico that serotype 1 and 2, we show lower vaccine efficacy compared to other serotypes, which could be in the main factor reducing the overall vaccine efficacy compared to other countries. But they're consistent among other countries, as you can see from the slides. This slide to show you the safety of it. The reactogenicity analysis shows similar adverse reaction reporting rates in both the vaccines as well as the control group. So the observed safety profile was acceptable during the 25 months of the follow-up in the over 13,000 vaccinees. So in conclusion, there was consistent result with the previous study. The primary outcome of the study was met with the vaccine efficacy of 60.8% against the symptomatic dengue caused by virology confirmed dengue caused by any serotype. Vaccine efficacy was observed against each of the 4 dengue serotypes. Vaccine efficacy against severe dengue cases due to any serotype after at least one dose was 95.5%. And additional exploratory analysis show an 80.3% reduction in the risk of hospitalizations due to dengue in the intent to treat population during the study. And vaccine efficacy was 83.7% in the seropositive participants. In the seronegative participant, efficacy was measurable but not statistically significant. Differences in vaccine efficacy by country may be explained by the differences in baseline serostatus and serotype circulation among the 5 countries. The safety profiles over the 25-month follow-up period is consistent with the favorable safety profile documented in previous study from phase 1 to 3. The results now available from 10 countries in Asia for CYD14 and Latin American CYD15 provide a consistent picture of efficacy and safety. The trials include populations with a range of dengue epidemiology, seasonalities, and level of endemicity. So CYD15 results suggest potential of this candidate dengue vaccine to provide important public health benefit in dengue endemic countries in both continents. So I'll just walk through quickly to compare the 2 studies, the results of the first 25 months active phase. This slide you can see the overall efficacy results, again the virology confirmed dengue due to any serotype and by serotype. So for CYD14 you can see it's 56.5 and for 15 will be 60.8. So this is after 3 doses of vaccine schedule. And the lower bound was significant. So in addition, results also show vaccine efficacy against symptomatic virology confirmed dengue of any severity caused by each of the 4 dengue serotypes, including serotype 2. This slide shows the overall efficacy and the intent to treat analysis. So show similar results. This was based on the 25 months follow-up starting at the first dose and confirmed vaccine efficacy against symptomatic biology confirmed dengue of any severity and any serotype. Okay, this is the intent to treat one. So the result for 14 was 70%. This slide will show the overall efficacy results for the virology confirmed dengue for the severe and hospitalized. So for the severe, there are two criteria we use: the IDMC, which are more comprehensive, including from the WHO 1997, as well as the Zero, which include all the severities of the dengue across assessment of severe dengue across study sites, more standardized. So we use both criteria. So as you can see that for vaccine efficacy in severe case by ADMC criteria is 70% in CYD14 and 95.9% in CYD15. And even by the WHO criteria shows 80% in CYD14 and 95% in CYD15 study. And for the reductions of the shown by vaccine efficacy of the hospitalized dengue cases, even quite consistent. For CYD14 is 67.2 and 80.3 in CYD15. There's a similar results for the safety profile observed between studies during the active phase, first 25 months, which is consistent with the previous report that identified with no major concern. So phase 3 efficacy results study in Asia and Latin America demonstrate a consistent efficacy and safety profile during the 25 months active phase. Both studies met their primary efficacy endpoints and show consistent safety profiles for the observed active phase. So both studies suggest the potential of these candidate dengue vaccines to provide an important public health benefit in dengue endemic countries. So I'll just go through the overall conclusions. This comes too close to my aim of the slide. So Sanofi Pasteur candidate dengue vaccine, a live attenuate tetravalent vaccine, has shown evidence of clinically efficacy against dengue in phase 3 clinical trials. Result of 2 large-scale phase 3 efficacy study, one performed in 5 countries in Asia, age range from 2 to 14 years, numbers over 10,000 children enrolled, and another 5 countries in Latin America and the Caribbean, age range from children and adolescents 9 to 16 years. The numbers of participants is over 20,000. Provide pivotal data on the efficacy, safety, and immunogenicity of this candidate vaccine. Primary endpoint was met in both studies. Overall efficacy against any symptomatic virology confirmed dengue was 56.5% and 60.8% respectively in children and adolescents who received 3 doses of vaccine. This is by per protocol analysis. And vaccine efficacy was observed against each of the 4 serotypes in both studies and varied from 34.7% in dengue serotype 2 to 80.9% in dengue serotype 4 during the 25 months of follow-up. The results suggest better protection in case of prior exposure to the dengue. Results show the reductions in the risk of hospitalization due to dengue by 67.2 and 80.3% in both studies respectively during the 25-month follow-up. Importantly, results also show efficacy against the severe form of disease, the DHF. During the 25 months of active phase of the 2 trials, the safety and reactogenicity profiles were consistent with previous report that identified no major concern, with similar reporting rate between the vaccine and the control group. These 2 large-scale efficacy studies show consistent safety and efficacy results during the first 25 months of follow-up of the active phase. So this will end my slide with the acknowledgement to the author and list of the CYD14 and CYD15 team. So across 10 countries nationwide, globally. Thank you.